This video discusses the genetics section of the biology syllabus. I previously did a video on Mendelian genetics, so genetics relating to Mendel's work with pea plants and that sort of thing, which was an introduction to the section of genetics. This is simply a continuation of that video. So if we ask the question simply, what is genetics? We know that genetics is the study of heredity and the variations that occur as genes are passed on through various generations. Genetics tries to explain both the similarities and the differences between parents and their offspring. So heredity is the passing on of traits or characteristics from one generation to the next. And genetic variation is the phenotypic and the genotypic differences between individuals of the same species, where the phenotypic differences are the physical features and the genotypic differences are the genes within the genome, the genes that control these physical differences. So your genotype would determine your phenotype. So we know in general that genetics is the study of traits and how they're passed on from one generation to the next, and the study of the variations of these traits, the variations in the phenotype and in the genotype. So genetics explains or tries to explain the similarities and the differences between parents and their offspring. Because we know that parents always produce offspring that look like them in some ways, but differ in other ways. So they look similar to parents, but not exactly identical. This is because half the heredity information in an organism is inherited from each parent via the gametes. So you're not going to look exactly like your mother because you've only got half her hereditary information. Your other half is from your father. So some of the experiments conducted by Mendel, we looked at in a bit more detail in the other video, but we know that Gregor Mendel was an Austrian monk. He was often called the father of genetics because he did a lot of work studying genetics. He was responsible for the first major breakthrough in the study of heredity by investigating the transfer of characteristics from one generation to the next. He did this looking at pea plants and at specific characteristics within pea plants. He realized that something is passed on from the parent to produce offspring, and that sexual reproduction combines these somethings from each parent to produce offspring which are unique yet the same. So they have some differences yet some similarities. But he didn't know what these somethings were. We'll later see that this has more to do with DNA and genetics and genes, but he didn't know. DNA hadn't been discovered when Mendel was doing his research. So in 1866, he published his work, but it wasn't really recognized in scientific circles until 1900. As Darwin's theory of evolution was gradually accepted, so were Mendel's findings and laws. So Mendel's work gave quite a valid explanation for Darwin's work, which was the theory of evolution. And Mendel's theory or his work on genetics kind of backed up Darwin's evidence. So a lot of other biologists also use Mendel's research as a basis for their own study, and Mendelian genetics is now a really, really important topic that's often studied and taught. We know that these somethings that Mendel talked about, these somethings that are passed on, combined in sexual reproduction and passed on to the next generation, are DNA. Well, the genes made up within DNA. The genes are found in chromosomes, and a section of DNA would simply be a gene. So in a cell, inside the cell, we know there's the nucleus. Inside the nucleus, there's the chromatin network or the chromosomes. Inside each chromosome, there's millions of strands or a strand of DNA. And within a section of DNA would be a gene. These genes are what are passed on from parents to children, to their offspring. So just comparing genes and alleles, we know that each chromatid, so each arm of the chromosome, these would each be a chromatid, is made up of one helical DNA molecule. Each DNA molecule is made up of a series of genes, so sections of DNA called genes. A gene is a section of DNA, so a series of nucleotides or a series of bases that controls hereditary characteristics, so that controls the traits that are passed on to the next generation. i.e., the gene is the basic unit of heredity in living organisms. Each gene occurs at a specific fixed location on a chromosome, which is called the locus of the gene, and each gene determines a particular trait in an organism. The difference between genes and alleles is that a gene may be, a gene's the more general term. So we know that a gene is a section for DNA, and a gene would determine a trait. For example, it would determine eye color. The allele is the specific variation of the gene. So an allele may be the gene for blue eyes. There could be another allele with the gene for, or there could be another gene. So the same gene, also the gene for eye color, but now it's the allele for brown eyes. We'll have a look a bit more at that later, but that's quite a basic knowledge that you guys should know by now. I've mentioned it in quite a few videos. So each gene has, or each chromosome has between several hundred to several thousand genes. The total number of genes in humans is between 20,000 to 25,000. So we have a lot of genes. Almost all somatic cells, so almost all body cells, have an exact copy of the genes in that organism. We say almost all cells because some may have a mutation. Because there are two of each kind of chromosome, so a paternal chromosome and a maternal chromosome, each cell contains two of each type of gene. And these different versions of the genes, as I said, are known as alleles. The gene pool is the set of all genes or the genetic information in a population of sexually reproducing organisms. So the gene pool is the set of all genes within a population. A population is a group of organisms of the same species found in a particular area. So they obviously have to be the same species, and they have to be found in the same area. So the gene pool of African elephants would be only talking about a specific population of African elephants. You wouldn't include the ones in Kenya and the ones in South Africa as part of the same gene pool, because it's only in a population. When there's a large gene pool, it indicates high genetic diversity and an increased chances of survival. So the large gene pool means there are more genes, more genetic information, more different alleles, which means that there's more genetic diversity and more genetic variation. Because of this, the organisms have an increased chances of survival because they have so much variation. They're able to adapt to different conditions, and it's a lot easier for them to survive in changing conditions. Whereas if they have a small gene pool with low diversity, there's a much higher possibility of extinction because they may not have the right variation that allows them to adapt to changing environments. And we know that the environment is constantly changing, especially with the current increasing rate of climate change. So they often ask you, they might ask about designer babies. I'm not sure if you guys have heard of them. Designer babies, another word for it is CRISPR babies, where basically it's kind of a little bit futuristic. It's not 100% happening at the moment, but the idea is that CRISPR is a gene editing tool that allows doctors or whoever it is, geneticists, to edit the genome of a cell. So that can possibly allow parents to get their embryo and then edit their embryo. So they say, or initially it's only been used for to treat genetic disorders. So to say like cystic fibrosis or something like that, the parents can check if the embryo has a possibility of having cystic fibrosis, and then they can use CRISPR to edit it so that the baby doesn't have it. But in the future, there's worries that people will take it from fixing genetic disorders to simply enhancing the baby's features. So for example, saying, okay, let's edit it to get a baby with blue eyes, brown hair, tall, smart, good at sport, and then they basically design their baby. It's a really controversial issue because of the possibility of moving on from solving genetic disorders to actually enhancing your offspring instead of just changing the life-threatening things about it. So you can Google CRISPR disorder if you want to. I mean, not CRISPR disorder. You can Google CRISPR babies, designer babies, if you want more information. They wouldn't really ask about it in paper 1. I don't think genetics is part of your guys' paper 2 model, but they could possibly ask it. I think you guys have reproduction, so they could definitely ask designer babies relating to reproduction as well. So it's quite a good essay topic. If you guys do want more information, you're welcome to message me, and I've got some extra details on designer babies, but don't stress about it, especially not for now. Maybe closer to the exams, but for now, just focus on learning the content. So how active are genes? Although each cell contains a full complement of DNA, so each cell has the entire genome within it, only the genes that are needed are activated within the cell, and the other genes are suppressed. Unlike in plants, which are totipotent, would mean that in the plant, every cell has the entire genome, and the entire genome is in its active form, so there are no genes or anything that are suppressed. So we know in humans and in animals, different genes are activated in different cells. These create the specific proteins that give the particular cell type its character. For example, bone cells, brain cells, skin cells, eye cells, etc. Some genes are active in many different types of cells, making the proteins that are needed for the basic functions of cells, and these would be called the housekeeping genes. For example, this might be a gene that produces an enzyme needed for cellular respiration, and obviously all cells undergo cellular respiration, so all cells would need this type of gene to be active. A high proportion of genes are non-coding genes, so they don't code for proteins and they occur in between the coding sections. They're sometimes called junk DNA, but we know that they're not junk. They do have a function. It's just not the same function as genes. If we look at the difference between homozygous and heterozygous, we also spoke about this in a previous video, but we know that after fertilization, the zygote will have 2 alleles for each gene, one from each parent. These will be in the same locus and the same location on each chromosome of a homologous pair. If the pair of alleles at the locus are the same, the organism is homozygous for that particular trait. If they're the same and they both code for the dominant trait, these would be homozygous dominant. If they code for the recessive trait, they would be homozygous recessive. If the pair of alleles at the locus are different, the organism is heterozygous, so it's a hybrid for that particular trait. And hetero means difference, homo means the same. That's basic English. So we can see here this cell is homozygous because the maternal chromosome and the paternal chromosome both have the same allele at the locus, whereas this is heterozygous because they have different alleles. We know that some alleles are dominant and others are recessive. In a heterozygous pair, one allele is dominant and the other is recessive. That's what heterozygous means. They're different alleles. If it's the dominant allele, it means the trait will be expressed in the offspring, which means that it does influence the phenotype of the individual. It masks the recessive allele, and when we represent it in a genetic diagram, we use a capital letter. If it's the recessive allele, the trait is suppressed, so it's not expressed in the phenotype of the offspring because it's masked by the dominant allele, and we represent this by a lowercase letter. So when we do genetic diagrams, the most common ones are Punnett squares, and they show how characteristics are inherited. So they show both the genotype and the phenotype of a cross between parents. The generations are always shown as follows, with P1 or just P being the parent generation, F1 being the first filial generation of offspring, so the first offspring from the parent generation, and F2 being the second filial generation of offspring, so the offspring of the first filial generation. In genetic diagrams, alleles can be shown as capital letters and lowercase letters, where obviously the dominant allele is capital letter, a recessive allele is lowercase letter. So as I said in the other video, try to choose letters where the capital letter differs from the small letter, so that you don't get confused in your genetic diagram as to which letter is actually the capital letter and which is the lowercase, and also so that your marker doesn't get confused. So something like S and S wouldn't work. C and C aren't good letters, but B and B are good. D is a good letter. F is a good letter. As there are 2 alleles for each characteristic, one on each chromosome of the homologous pair, so the maternal allele and the paternal allele, the 2 letters are written for each characteristic. So if we're talking about the height of plants, we could use, where tall is the dominant trait, we could use a letter T. For capital T, obviously for the dominant trait, so tall, and lowercase t for the recessive trait, so short. And this is how you'd write the 3 different offspring. So homozygous tall, homozygous dominant, homozygous short, so homozygous recessive, and heterozygous, which we know has a dominant and recessive allele, but the dominant trait is obviously the one shown, so the plant would be tall. If a plant has, or if an organism has complete dominance, that means that the characteristic that is fully expressed in the phenotype of a heterozygous organism is the dominant allele. When a dominant and recessive allele are present together, only the dominant allele has an effect on the phenotype. That we know that from looking at just heterozygous organisms. So the heterozygous phenotype looks or is the same as the homozygous phenotype. The offspring that is heterozygous has the same appearance as the offspring that is homozygous dominant. The characteristic that is masked, so in this case shortness, when the organism is heterozygous is the recessive allele, and it's only expressed when it's homozygous recessive, so when there's no dominant allele at all. We know that Mendel's laws of dominance says that in a cross of parents that are pure for contrasting traits, so one parent is pure for the dominant trait, so homozygous dominant, the other is pure for the recessive trait, so homozygous recessive, only the dominant trait will appear in the phenotype. So all the offspring will be heterozygous, and they will all have the phenotype of the dominant trait, obviously because we know that in heterozygous offspring, the dominant trait is expressed. The recessive allele is masked. So then if we move on to sex chromosomes, we know that somatic chromosomes had a diploid number of, somatic cells had a diploid number of chromosomes. Now gamete cells have, or sex cells have a 23 chromosomes, so a haploid number. Those are sex cells. If we look at sex chromosomes, they aren't the cells, they're actually the chromosomes of the 23rd pair. In a gamete, so in a sex cell, they'll obviously only have one sex chromosome, whereas in a somatic cell, they'll have two sex chromosomes. And these two sex chromosomes determine the gender of the offspring. So of the 23 pairs of chromosomes in a human cell, 22 pairs have more or less the same shape, so they're homologous, and these are called autosomes. The 23rd pair differ in shape, and these are the sex chromosomes, also called the gonosomes. As I said, they determine the sex of the offspring. The sex chromosome that is longer is the X chromosome, and the shorter one is the Y chromosome. In a female cell, or a cell that in a female's body, has two X chromosomes, so they are homogametic, and they have the genotype XX. Males have one X chromosome and one Y chromosome, so XY, and we say that they are heterogametic. We can see that the X chromosomes are longer, whereas the Y chromosomes are a lot shorter. If the sex chromosomes are XX, this means that they are going to be inside the cells of a female organism. That means that the undifferentiated sex organ, so the gonad tissue within the embryo, it's going to develop into an ovary, and the fetus will become a female. If the sex chromosomes are XY, the gonad tissue will develop into a testis, and the fetus will grow into a male. For sex determination in humans, the sex of an organism is determined by the presence or the absence of a Y chromosome. So if the Y chromosome is present, it's a boy. If the Y chromosome is absent, it's a girl. This switch occurs when the embryo is 7 to 8 weeks old. That's when the gender is kind of determined. After this, many other genes, mainly on the autosomal chromosomes, so the ones in pairs 1 to 22, they control the detailed differentiation of the male and female characteristics of the fetus. The inheritance of sex is a special form of monohybrid inheritance. We know that monohybrid inheritance is when a trait is determined by only one gene. So now, obviously, it's not just a single gene, but it's an entire chromosome. But it's the same process where you can use a Punnett square to determine the sex of an organism. So you can work out sex inheritance in just the same way as for any other characteristics by using the letters X and Y instead of a capital letter and a lowercase letter. And these letters obviously describe whole chromosomes instead of individual alleles. So if we looked at, obviously it's a male and a female undergoing reproduction, the female's here, so they're XX, and the male is XY. Together they can have an XX and an XX, or an XY and an XY. So basically they have a 50% chance of producing a female, 50% chance of producing a male. And obviously that's true. You're not going to have a 75% chance of producing a girl. It's always 50-50 for males and females. Sorry, males and females. If we just look at a few questions, the type of cell division that results in gametes is obviously meiosis. If a normal human cell has 46 chromosomes, how many chromosomes are there in a human sperm cell? That's obviously 23, the haploid number. In a human boy, which sex chromosome came from his father? That's the Y chromosome. And which sex chromosome came from his mother? That's the X chromosome. And we can see here that that's true, where this would be the mother, and this is the father's sex chromosomes. For example, this is the human boy we're talking about. We can see that the X came from the mother and the Y came from the father. We know that in the formation of an embryo, every embryo has an X chromosome. The other one is either an X or a Y chromosome. So a female always gives her X chromosome. You always have your X chromosome from a mother, and your other chromosome, which could be an X, comes from your father. If it's a Y, it also comes from your father. Of the two sex chromosomes, which one carries the largest number of genes? And we know that that's the X chromosome because it's much longer in size. There's also some alleles that are sex-linked. The Y chromosome is very short and has very few alleles on it, other than those responsible for maleness. But the X chromosome is quite a bit longer. It can carry many alleles along its length in addition to those for femaleness. So only a very small part of the X and Y chromosomes can pair up during meiosis. They have complementary genes because obviously if you look here and the way this is an X chromosome and this is a Y chromosome, these parts could be homologous, they could pair up, but this obviously doesn't have any complementary pairs because there's nothing to pair it to. So no crossing over occurs between the X and the Y chromosomes during meiosis. The alleles that are carried on the non-homologous part of the X chromosome, so this green part here, they're called X-linked genes or sex-linked alleles. How are sex-linked alleles inherited? We know that men only have one X chromosome, so they'll only have one of each sex-linked allele, which is carried on the X chromosome. Women have two X chromosomes, so they'll have two sex-linked alleles, one on each of their two X chromosomes. The alleles on the X chromosome, therefore, can only be inherited by either sex, but the male only receives one of the possible alleles because it only has one X chromosome. The female has two X chromosomes, so it receives two sex-linked alleles, which means that the female can be homozygous or heterozygous. Some examples of sex-linked diseases are a cleft palate, diabetes insipidus, red-green color blindness, hemophilia, and muscular dystrophy. There's more than 120 sex-linked diseases. Certain genetic diseases seem to occur more often in males than in females. Why is that? That's because the male only has one X chromosome. So if a gene mutates, or if a gene for a disease is present on the X chromosome, then the male will get the disease, even if it's the recessive allele, because the male only has one X-linked chromosome. If it gets that allele for the disease or for the recessive trait or for the mutation, it's 100% definitely going to show it because there's no way that it can be masked because there's no other allele for it. Whereas in a female, even if a female does inherit one recessive trait or the trait for a disease, it has a possibility of being masked if on her other chromosome she has the dominant trait or the trait that that's not for the disease and that can mask the other trait. So the female can only express the traits of the recessive allele if she has two recessive alleles, so then there would be nothing to mask it. So the chances of the female getting the disease are less because they have to be homozygous recessive. If they're homozygous dominant or heterozygous, they won't show the disease, obviously, unless it's a dominant disease, but that's a bit more uncommon. We don't talk about that as often. But if you're heterozygous as a female, and even if you're not showing the disease, you can still pass it on to future generations because obviously you have the allele for it anyways. That means that you are called a carrier. So some of the recessive sex-linked alleles that are found on the X chromosome would be those for red-green color blindness and those for hemophilia. So red-green color blindness, We have genes that produce photopigments in the cones in the retina of the eye, and these are carried on the X chromosome. These photopigments are sensitive to different wavelengths of light, which allows us to see color better. If the genes are missing or if the genes are damaged, then certain photopigments are affected. More than 99% of all colorblind people suffer from red-green color blindness, so it's the most common form of color blindness. An individual with this form of color blindness finds it difficult to tell the difference between various hues of red and green. The genes for red-green color blindness is transmitted from the colorblind male on his X chromosome to all his daughters, who then become heterozygous carriers and are usually unaffected. That's if the female doesn't have the gene for red-green color blindness. So obviously this male son doesn't get the recessive gene because they get his Y chromosome. On his Y chromosome, there are no genes for color blindness, so they can't inherit the trait. So only mothers can pass on a disease only mothers can pass on the actual disease as opposed to being a carrier. Fathers can pass on the recessive gene, but they can't pass on the actual disease because the daughters would get the chromosome from their mother that masks this trait. If the mother has it, if the father has it and passes it on to his daughters, and the mother has the disease, so she's homozygous recessive, she'll definitely pass it on to her daughter as well, and her daughter will be red-green colorblind. If the father has it and the mother is heterozygous for it, she may pass it on to her daughter, which means that her daughter can either be homozygous recessive, so show the disease, or if she doesn't pass it on to her daughter would be heterozygous, so a carrier. So the sons of an affected male won't inherit the trait from them because they only inherit his Y chromosome. So we can see that if we look at an unaffected mother and an affected father, the mother has 2 dominant alleles, whereas the father has only 1 allele, which is recessive. For their first son, they have 4 possibilities of children that they can produce. For the first possibility is a son that doesn't have color blindness because he received his father's Y chromosome and his mother's unaffected X chromosome. They can also have a chance of producing an unaffected son with its father's Y chromosome and its mother's other chromosome, but because she's homozygous dominant, the other chromosome is obviously also dominant, so their son also won't have the disease. But if we look at the daughters, they'll receive their father's affected X chromosome and their mother's unaffected Y chromosome, either one, creating 2 different daughters, but obviously with the same characteristics. So both their daughters will be heterozygous for the disease. Or when I say both their daughters, they have a 50% chance of producing a daughter, and it's guaranteed that their daughter will be a carrier because their father's affected. A carrier woman has a 50% chance of passing on a mutant X chromosome to each of her sons because her son she can pass on one of her 2 X chromosomes, and the 1 chromosome is unaffected whereas the other is affected, so that makes it a 50% chance. So if we look at a father that is unaffected but a mother that is affected, or she's not affected, she's a carrier, sorry. So she has 1 normal allele, 1 dominant allele, and 1 defective allele, so 1 recessive allele. The daughter will receive its father's X chromosome, and she could receive her mother's dominant allele, which means that she's normal and she's unaffected by the disease. She can also receive her father's X chromosome, but her mother's affected X chromosome, meaning that she's heterozygous. She doesn't have the disease, but she's a carrier. The son will obviously receive his father's Y chromosome, and he could receive his mother's X chromosome, the unaffected X chromosome, which means that he's unaffected by the disease. The son could also receive the father's Y chromosome, but the mother's affected X chromosome, meaning that he would be affected by the disease, because the only allele he has is that for the trait of the disease. So is that for the recessive allele, which means that he's going to show the traits of the disease. There's nothing to mask it. A female will only be colorblind if there's a recessive colorblind on each of her 2 chromosomes. So she's homozygous recessive, and that's quite rare. Should an affected male have children with a carrier or a colorblind woman, their daughters may be colorblind by inheriting an affected X chromosome from each of their parents, which means that their daughters will be homozygous recessive. If we look at some quick questions on which chromosomes are most sex-linked genes carried, we know obviously this is X. And what is a character? So to define a carrier, we know that a hereditary carrier is a person or other organism that has inherited a recessive allele for a genetic trait or mutation, but usually does not display that trait in their phenotype or show symptoms of the disease because we know it's masked by the other allele. It's quite important to be able to do this as a Punnett square or genetic diagram. So use the letters B to represent normal vision and lowercase b to represent color blindness in the table below. In the table below, fill in 5 possible genotypes and the corresponding phenotype, so physical characteristics, representing both normal vision and color blindness in males and females, using the symbols X and Y to show the gender. So this is basically a table that shows us the 5 possible variations of phenotype that can occur. In doing sex-linked questions, it's just one step further from normal Punnett square genetic questions, where you have an X and a Y, or an X and an X, to show the sex of the organism, but then you also have a capital letter or a lowercase letter to show the presence or absence of a trait. So a Y chromosome is obviously just a Y chromosome. It won't have a lowercase or capital letter because it's an X-linked allele. It's not present on the Y chromosome. Then there's 2 types of X chromosomes, the one that has the dominant allele and the one that has the recessive allele. So there's 2 types of males. It can either be a normal male, so the one with the Y chromosome and a dominant allele X chromosome, or a colorblind male, so a Y chromosome and a recessive X chromosome. So the 5 different types of genotypes can be, as I said, those 2 males that I said here, and in the females, it can be a homozygous dominant female, so 2 X chromosomes to show it's female, 2 capital B's to show that it's normal. She can be a carrier, so 2 X's for female, a capital B for the 1 dominant allele, and a lowercase b for the recessive allele. She won't show the trait in her phenotype, but she's a carrier. And we can have a homozygous recessive female, where there's 2 lowercase letters, as she has 2 recessive alleles. For the genotypes, if the male has a dominant allele, he's a normal male. If he has the recessive allele, he'd be colorblind. If the female has 2 dominant alleles, she's a normal female. If the female has 1 dominant allele, 1 recessive allele, she's a carrier female. She doesn't show the symptoms, but she carries the trait. If she has 2 recessive alleles, she's a colorblind female. And you could write it out in a Punnett square like you do any other normal traits, where they'd give you the parents' genotypes, and you'd have to figure out the offspring genotypes. If you're struggling to do a Punnett square example like this, you're welcome to send me a message, and I can explain it to you, or I can check you're working. Hemophilia is another example of a sex-linked disease. It's a rare genetically determined condition of frequent excessive bleeding because the blood clots very slowly. There's a dominant allele H, which allows the blood to clot. In this case, it's a good thing. We want the blood to clot. If it doesn't clot, we have excessive bleeding. And we have a recessive allele H, lowercase h, that doesn't allow the blood to clot normally. So the recessive allele is that gives the offspring hemophilia. The alleles controlling blood clotting are sex-linked alleles located on the X chromosome, so the same as the alleles controlling red-green color blindness. There's no gene for blood clotting on the Y chromosome. For the male genotypes, there are only 2 possible genotypes for the male, because we know that the Y chromosome doesn't have a gene to code for hemophilia. So the male can either get the dominant allele on the X chromosome, which means that it's a normal male, or if the male inherits the X chromosome with a recessive allele, he will be a hemophiliac. So hemophilia is caused by a recessive allele on the single X chromosome. For females, there are no known female hemophiliacs, So there are no known women who are homozygous recessive. We think that this condition causes a natural abortion. But women can be carriers of the recessive allele. Because of that, there's only 2 possible genotypes for a female. Obviously, in general, there are 3, including this one, but this one doesn't count because there's no living females that have this genotype. So the only 2 possible genotypes would be homozygous dominant, making it a normal female, or heterozygous, meaning that the female appears normal, but she is a carrier. Hemophilia, we therefore say, is a male disease because males are the only gender that will show symptoms of the disease. And then moving on from sex-linked inheritance, the last little point is polygenic inheritance. So some simple traits like eye color is caused by just one pair of alleles. So just a single allele on the maternal chromosome and a single allele on the paternal chromosome. But for some traits, there are more than one pair of alleles that are responsible for the trait. An example of these traits would be skin color. So they're usually more complex traits, and they're determined by the interaction of many different alleles. Each allele has a small individual effect on the offspring, and this results in a range of phenotypes, as opposed to one single or 2 different phenotypes. For example, the presence of dimples could be a monogenic inheritance, non-polygenic, because you either have dimples or you don't have dimples. But for skin color, it's polygenic. There's a whole range of skin colors. You can be very light, you can be slightly light, you can be very dark. As we can see, there's a whole range. So instead of having just like AA, just 2 different alleles coding for it, you now have a multiple alleles coding for it. So multiple different pairs of alleles, and you'd need to use multiple different letters to show it. But they won't ask you this in a Punnett square or anything. It's just for extra knowledge. What is meant by continuous variation? The more pairs of alleles that control a characteristic, the greater the number of possible combinations, obviously, and the greater the variety of the phenotypes, which is what we spoke about here in the range of the phenotypes. So the more variations of that phenotype or genotype, the more alleles there are for that characteristic. This is just inverting this statement. Each phenotype will differ slightly from the next, which forms a graduated series, so a whole range. And this is what continuous variation is. Continuous variation is the graduations of a characteristic in a phenotype. Some characteristics that have continuous variation would be height in humans, skin color, metabolic rate, longevity, hair color, etc. So that is the introduction to genetics. I will do another genetics video in the next few days, but if you have any questions, just let me know. It's quite important to be able to draw a Punnett square or a genetic cross. I know that some schools do use genetic crosses. Don't stress about that. If I keep talking about Punnett squares and I don't mention genetic crosses, it's simply because I didn't do genetic crosses at school. I only did Punnett squares. But they'll very often ask you Punnett squares or genetic crosses, and it's often up to 6 or 7 marks. So it's quite important to know how to do it. I did a bit more information on how to draw them in the previous video on Mendelian Genetics. So have a look at that if you're struggling. And you're welcome to message me and ask any questions on that or on anything else.